Hello everyone. Today we are going to talk about nephrology, which includes a basic introduction on kidneys, its parts, and some of the functions that are dealt with kidneys and nephrons. The study of kidney in healthy and diseased conditions is called as nephrology, and the kidneys are bean-shaped paired organs lying on either side of the spine in the upper part of the abdomen. The kidneys are retroperitoneal, which means they lie behind the peritoneum, the lining of abdominal cavity. They are approximately at the vertebral level T12 and L3, and the right kidney usually lies slightly lower than the left in order to accommodate liver. Here, T12 stands for thoracic vertebrae 12 and L3 stands for lumbar vertebrae number 3. Proceeding further, each kidney is connected to the aorta and vena cava by a single short wide renal artery and renal vein. By means of these blood vessels, kidneys receive a blood flow of around 1200 milliliters per minute, which represents around one-fifth of the cardiac output at rest. In a normal human adult, each kidney is about 11 centimeters long and about 5 centimeters thick, weighing 150 grams. If you could see the diagram, here is the kidney, and you could see the parts that are labeled here, such as cortex, medulla, calyx, pelvis, renal artery and renal vein, papilla, renal column, pyramid, and the ureter. Nephron. Each kidney consists of approximately 1 million microscopic functional units called as nephrons. The basic function of the nephron is to regulate water and soluble substances in the body by filtering it out first, reabsorbing what should be kept and excreting the rest. The nephron is made up of two components: glomerulus surrounded by a hollow bulb known as Bowman's capsule and a tubule. From this diagram, you could see the mesh kind of structure that you see here of blood vessels is called as glomerulus, which is followed by Bowman's capsule, and the tubule, which is again divided into a proximal tubule, loop of Henle, and distal tubule, which will be followed by a collecting duct. Glomerulus is a capillary bed found surrounded by the Bowman's capsule. The glomerular capillaries form a network of parallel paths from the feeding afferent arteriole to the efferent arteriole, which drains blood from glomerulus and splits again to form peritubular capillaries in the cortex and vasa recta in the medulla. This organization of circulation is unusual as blood leaving capillaries passes into the arteriole as opposed to the venule. The resistance of arterioles result in high pressure in the glomerular capillaries, aiding the process of ultrafiltration, where fluids and soluble materials in the blood are forced out of the capillaries and into the Bowman's capsule. Fluids are collected in the Bowman's capsule is known as glomerular filtrate, which eventually becomes urine after further processing along the nephron. Here, the highlighting words that you should remember are the ultrafiltration, which is the process of the process that happens inside the glomerular capillaries, and the other one that you have to remember is the glomerular filtrate, which is basically the filtrate substance that is collected in the Bowman's capsule. Continuing with glomerulus, the process of filtration of blood in the Bowman's capsule is called as ultrafiltration or glomerular filtration. The normal rate of glomerular filtration is about 125 milliliters per minute, which is equivalent to 10 times the blood volume every day. The glomerulus acts as an ultrafilter and separates water and cholesteroloids from the blood plasma while retaining blood cells and proteins. Any proteins under the Dalton size can pass freely through the membrane, although there is some extra hindrances for the negatively charged molecules due to negative force of the basement membrane and the podocytes. Any small molecules such as water, glucose, salt, amino acids, and urea pass freely into nephron, but cells, platelets, and large proteins do not. Bowman's capsule leads into a long convoluted tubule consisting of four sections. Proximal tubule twists into a tiny knot and a loop of Henle, distal tubule, and a collecting duct. The collecting ducts empty into the central cavity of the kidney, the renal pelvis, which connects to the ureter. Proximal tubule reabsorbs fluid in bulk from the filtrate, including two-thirds filtered salt and water and all filtered organic solutes. This is driven by sodium transport from the lumen into the body by sodium potassium ATPase in the basolateral membrane of the epithelial cells. Much of the mass movement of water and solutes occur in between the cells through the tight junctions that in this case are not selective. The solutes are absorbed isotonically in that osmotic potential of the fluid leaving the proximal tubule is the same as that of the blood. However, glucose and amino acids are absorbed actively via cotransport channels driven by the sodium gradient out of the nephron. The loop of Henle descends from the cortex into the medulla and then ascends back from the medulla into the cortex only in juxtamedullary nephrons. In other nephrons, the loop merely extends through the kidney's cortex. The longer the nephron, the better it performs, and its counter-current exchange mechanism involving the loop of Henle in the medulla allows reabsorption of water and salt. By this point, only about 6% of the original water and 4% of the original salt remains. The distal tubule is similar to the proximal tubule. Salt is pumped out and water follows by osmosis. After traveling through the distal tubule, only 3% of the water remains and the remaining salt content is negligible. A third process, in addition to the filtration and reabsorption, which takes place is called as secretion. Hydrogen and potassium ions are actively transported from the blood into the urine to be excreted. The collecting duct goes deep into the medulla and as much as 3/4 of the remaining water can be reabsorbed as the urine travels through the collecting duct. But in reality, the amount of water reabsorbed is dependent on the permeability of the walls of the duct, regulated by ADH, which stands for antidiuretic hormone, secreted by the posterior part of the pituitary gland. The more ADH, the more permeable ducts and the more water reabsorbed. The less ADH, the less the permeable ducts and the less is the water reabsorbed. This is related to the high osmosis or osmolarity of the interstitium due to the countercurrent multiplier system of the loop of Henle. Dehydration results in an increase in ADH, while water sufficiency results in low ADH following for diluted urine. Lower portions of the collecting duct are also permeable to urea, allowing some of it to enter the middle of the kidney, thus maintaining its high ion concentration, which is very important for the nephron. Here you could see a brief glimpse on a basic block diagram or a basic functional diagram of a nephron, as in where vital substances are secreted, absorbed and filtered out of the body. Some of the major functions of the kidney include removal of waste products through urine, regulation of water and electrolyte content of the body, maintenance of the chemical balance of the body, retention of substances vital to the body such as protein and glucose, and also help in making hormones that control blood pressure, making of blood cells, and maintenance of bones. Filtering waste from bloodstream involves in several steps, as you might have just understood from the figures. It involves glomerular filtration, Bowman's capsule, loop of Henle, the ducts and the tubules, and also some parts of secretions. So wastes are filtered out from the blood in the glomeruli via the process of ultrafiltration. The ultrafiltrate is passed through, in turn, the proximal convoluted tubule, the loop of Henle, and the distal convoluted tubules, and is then collected by the collecting ducts to form urine. The renal collecting ducts open into the renal pelvis and drain into the ureters, which pass on to the urine to the bladder. Maintenance of body sodium and water balance. There is a stable balance of sodium and water in the body. The major homeostatic control point for maintaining this stable balance is renal excretion. The kidney is directed to excrete or retain sodium via the action of aldosterone, ADH, ANP, which stands for atrial natriuretic peptide, and other hormones. Chloride always follows sodium. Acid-base homeostasis. Metabolic reactions are very sensitive to pH level of the fluid in which they occur. This is because of hydronium ion concentrations that can influence the enzyme functions. The kidney maintains blood plasma acid-base homeostasis by hydronium ion regulations. Gain and loss of hydronium ion must be balanced. The sources of hydrogen ion or H plus ion are carbon dioxide, production of non-volatile acids from the metabolism of proteins and other organic molecules, gain in hydrogen ions due to loss of bicarbonate in diarrhea and non-gastric GI fluids, gain in hydrogen ion due to loss of bicarbonate via urine, and hyperventilation. On the contrary, the sources of hydrogen ion loss are hyperventilation, hydrogen ion loss via urine or via vomitus, and also via metabolism of various organic anions. When hydrogen ion loss exceeds gain, alkalosis occurs. When gain exceeds loss, acidosis occurs. There are various renal responses to acidosis and alkalosis. For example, a kidney responds to acidosis by adding bicarbonate to the blood plasma via tubular cells. This is caused by sufficient hydrogen ion secretion from the tubular epithelial cells. Extra hydrogen ion secretion will bind to the non-bicarbonate urinary buffers, and this will lead to more new bicarbonate in the blood plasma. This is also caused by increased glutamine metabolism and ammonia excretion. And in response to alkalosis, kidneys try to excrete bicarbonate via urine. This is caused by lowered rate of hydrogen ion secretion from the tubular epithelial cells, and this is also caused by lowered rates of glutamine metabolism and ammonia secretion. Coming to the secretion of hormones, there are several important hormones that are vital to the body functions that are operated by kidney. For example, secretion of erythropoietin, which regulates red blood cell production in the bone marrow. You could remember it very well because erythro is almost anonymous, and almost analogous to red blood cells. So erythropoietin refers to red blood cell production, and secretion of renin, which leads to the formation of angiotensin 2, which is responsible for controlling of blood pressure. Secretion of active form of vitamin D, which is 1,2,5-dihydroxy cholecalciferol, which is also called as calcitriol, and prostaglandins, which are helpful in maintenance of bone health. So this is a basic introduction on kidneys, nephrons, and basic functions of each of them. I hope you could remember and recollect pretty soon.